So catabolic reactions and anabolic reactions work together all the time inside living cells. Again, the catabolic reactions release energy by breaking molecules, and then that energy can be used to power anabolic reactions. So in other words, by breaking down something like a carbohydrate, like glucose, into CO2 and water, that can provide the energy to build amino acids into something big like a protein. The energy released by catabolic reactions can't jump directly into the anabolic reactions. They need something to physically transfer that energy from one set of molecules into another set of molecules. So there are actually a couple of extra steps that happen inside living cells. When these big molecules are broken down and that energy is released, they will often be used to build some smaller molecules, things like ATP and some of our molecules known as energy carriers like NADH and FADH2. Those small molecules will then be transferred into the anabolic reactions. They'll be broken down, which is another tiny catabolic reaction, and then that energy will be released into the reaction that can build something big, something like a protein, for instance. Now, once that happens, those little energy carriers are going to be depleted. So they're going to need to travel back to the catabolic reactions so they can pick up some more energy and be built again, which is another little anabolic reaction. They can then dump their energy out into the anabolic reactions again to build big things. They get depleted, and so on. So we have this cycling of energy between large molecules and small molecules that keeps everything running. Next, let's look at a few specific examples of catabolic and anabolic reactions. First, we have a set that we've talked about before, oxidation and reduction. Oxidation and reduction reactions produce ions. So they have to do with the transfer of electrons from one molecule to another. In an oxidation reaction, a molecule is going to lose an electron. So this little green compound, whatever it is, has lost an electron and it's gained a positive charge because of that. So we say it has become oxidized. Now, once it loses one or more electrons, it's going to have that positive charge, so we would call it a cation. Reduction is the opposite. In a reduction reaction, a compound is going to gain an electron. So this little orange blob over here is taking in that electron, or sometimes more than one electron. So we say it has become reduced. Because it's been reduced, it's going to have a negative charge, and we would call it an anion. Now, I know the word reduction seems kind of backwards. It's reduced, which makes you think it's getting smaller, but it's actually gaining an electron. So you just have to remember that it's one of those opposite words in biology. It's a poorly named term, especially for freshman students. What's actually being reduced when this molecule takes in that electron is the amount of potential energy that it has. So that's why it's called a reduction. Here's a memory trick that you can use for oxidation and reduction. I'm from North Texas, and there's a lot of oil production, a lot of oil rigs going on up there. So when I think of oxidation, I think of the word oil, which stands for oxidation is losing. And then I think about rig for reduction. And rig stands for reduction is gaining. What's being lost and gained? Well, electrons, of course. But remember, oil rig. Oxidation is losing, reduction is gaining. Oxidation and reduction reactions go together. When one thing loses an electron, something else is going to take it up. The cell doesn't typically just release electrons into the environment because they can cause a lot of havoc. When oxidations and reductions go together, we refer to that collectively as a redox reaction. And here's an example. When we put sodium and chlorine atoms together, sodium is going to give its electron to chlorine. Sodium, in this example, goes through an oxidation. It is oxidized. And chlorine goes through a reduction. It is reduced. Sodium gets a positive charge because it's lost an electron, and chlorine gets a negative charge because it's gained an electron. Now, this particular redox reaction is pretty easy to interpret because there's a complete transfer of electrons from one atom to another, and that's represented with these little symbols. Other redox reactions can be a little trickier to recognize because sometimes in these reactions, the electrons will just be shifted around a little bit, and it changes the potential energy within the atom and within the molecule. It's important to remember that reactions like oxidations and reductions represent a transfer of energy from one molecule to another. For instance, in an oxidation, an electron is going to be lost. That electron carries energy with it. So when that electron is transferred from one molecule to another, it carries a little bit of energy away from the original molecule, and it adds it to the second molecule. Now, electrons can have different amounts of energy depending on what's going on with them. If the molecules are exposed to heat or light, their electrons tend to have more energy than if they are cold and sluggish. Another category of energy carrier molecules are known as electron carriers. These include NADH and FADH2. Now, unlike ATP, which is dephosphorylated and phosphorylated to release and gain energy, these guys work by being oxidized and reduced. They work with electrons specifically. So let's look at an example. Here we have NAD+. You can tell that NAD+ has been oxidized because it has that plus sign. The plus sign indicates that it's lost an electron. Now it's going to want to get that electron back. So how can it do it? Well, to do this, it needs to interact with two hydrogen atoms. From one of the hydrogen atoms, it's going to steal an electron. Poor little hydrogen gets picked on all the time. It can then bind with the other hydrogen atom, forming NADH. This is the reduced form. Now, the reduced form has more energy in it than the oxidized form. So that reduced form can travel somewhere else in the cell, get broken down again, and then those energized electrons can be used in other reactions. They carry the energy necessary for that reaction to do its work. So this cycle repeats over and over again. Another way to think of these electron carriers is either as a taxicab or as a dump truck. I like the dump truck, but let's look at the taxicab. Here we have a taxicab that is empty. This is the oxidized form, NAD+. When it's loaded with passengers, it's full of energy. It's the reduced form. It can then travel somewhere else in the cell and dump its passengers out, becoming oxidized again. I like to think of these as dump trucks because I like to picture little dump trucks traveling to maybe a coal mine, getting filled up with energy from the coal mine, and then traveling somewhere else in the cell, becoming oxidized and dumping out their coal to energize other reactions. But either analogy works. This chart shows the oxidized and reduced forms of our electron carriers. The oxidized forms have less energy in them than the reduced forms. The reduced forms have more energy. That's the energized form. So for instance, NAD+, after it interacts with two electrons and its two hydrogens, it will form NADH and a hydrogen ion. This is the form that holds the most energy. It can then travel somewhere else in the cell and get oxidized back into NADH. It will release those two little electrons with their energy into another reaction to do some work. Another energy carrier you will see is FAD, FADH2. The oxidized form that's empty is FAD. It will also interact with electrons and hydrogens to form the reduced form, the energized form, which is FADH2. This energy carrier we will see in photosynthesis, so just kind of keep it in mind for now, but we'll see it in action in Chapter 10. This year turn represents a reaction that we're going to look at in more detail in Chapter 9, and it is purposely tricky. In order to answer these questions, you really have to think about what you know about oxidations and reductions, and you need to look at the diagram and interpret what you see. See what answers you could come up with and bring them to class with you, and we'll discuss it. Another set of reactions you're going to see in action quite a bit in later chapters are phosphorylations and dephosphorylations. What fantastic long words. To put it simply, a phosphorylation occurs when a phosphate group is added to something. So if we have this blobby protein here, and we add to it a phosphate group, we say it has been phosphorylated. By adding this big old phosphate group to that molecule has gained some energy. So phosphorylation represents an endergonic anabolic reaction. The opposite of that is known as dephosphorylation. This is where you take a molecule that has a phosphate group and you remove it. You take the phosphate off. Dephosphorylations release energy. So at the end of the reaction, the product has less energy than the reactant that you started with because that phosphate group carries energy. This is considered an exergonic or catabolic reaction. Let's talk about ATP in a little more detail. ATP is by far the most important energy molecule inside your cells. It's like the energy money or the energy currency of your cells. Anytime your cells want to perform a little chemical reaction that requires a little energy, it will break down an ATP. And then that ADP has to be recycled. This is accomplished through a series of phosphorylations and dephosphorylations. So let's start up here with ATP, adenosine triphosphate. It has 3 phosphate groups, and that is where the energy is stored. Now, if your cell wants to perform a chemical reaction, it's going to need to harvest some energy out of this ATP. It's going to do that by dephosphorylating it, by removing a phosphate group. So the leftovers there are ADP, or adenosine diphosphate, and a lone sad little phosphate group. That releases energy that can do work. Now we need to recycle this ADP back into ATP. So what can we do? Well, we're going to put that little phosphate together with the ADP, forming ATP again, capturing that energy inside ATP. Glucose metabolism, or cellular respiration, which we're going to get into in more detail in a minute, is largely about this part of the cycle, about taking ADP, adenosine diphosphate, and phosphorylating it. Where does the energy come from for that? Well, that's the breakdown of glucose. Energy is funneled out of glucose and used to build as much ATP as possible. So the hydrolysis of ATP into ADP is an exergonic reaction that releases energy that can be used to do cellular work. But how does it do that? Well, to understand how the hydrolysis powers different reactions, you have to think about these phosphate groups. Remember that the phosphate groups are not just a phosphorus floating around in space. A phosphate group is a fairly large functional group. It's a phosphorus atom connected to 4 oxygen atoms. So that functional group carries a lot of bond energy with it. And when that group is transferred from one molecule to another, it has the effect of either energizing that new molecule or changing its shape or making that molecule more reactive than it was previously. And this can push that molecule into the reaction that we want. The hydrolysis of ATP into ADP can be used to drive chemical reactions. We refer to this as doing chemical work. In this example, the phosphate group from this ATP has been removed and attached to whatever this purple molecule is here. That makes the purple molecule a now phosphorylated intermediate, less stable than its beginning form. And so it allows it to enter into this reaction with whatever this green square molecule is. At the end of the reaction, the product is a fusion of the purple triangle and the green square, and you see that the phosphate group has been removed a second time and released to make room for that bond. The hydrolysis of ATP into ADP can also be used to do mechanical work. In other words, it can be used to make molecules physically move relative to one another or change shape. In this example, ATP is interacting with two proteins that are responsible for muscle contraction. We've got this big guy here called myosin, and this little chain of pearls that should look a little familiar, that's actin. And they interact with each other to make the muscles contract. What's happening is that a phosphate group has been removed from ATP and instead has been attached to these proteins at the junction where they meet. That's going to make them change their shape relative to one another, and it makes the myosin actually pull on the little actin fibers, so they kind of slide past one another, and that's what allows the muscle to get shorter or actually contract and produce force. What are we left over with? Well, when we're all done, that phosphate will be removed again from that junction, and so we're left with ADP and inorganic phosphate, also known as PI, and that ADP will have to be recycled back into ATP again. ATP energy can also be used to help drive the transportation of specific molecules across the plasma membrane. In this case, the phosphate group from an ATP has been removed, it's been hydrolyzed, and it's been attached to this transport protein. Now remember that proteins can change their shape, so the addition of this phosphate group to the protein is going to make it change its shape. And in that case, it's going to allow it to pick up some more of these solutes. The later detachment of this phosphate group, the dephosphorylation of this protein, is going to make it change its conformation again, which will allow it to release those solutes onto the other side of the membrane. So this little protein does this over and over again. As it's phosphorylated, using the phosphate from the ATP, it changes its shape. When it's dephosphorylated again, it changes its shape again. And so that's how these proteins can actually pump things from one side of the membrane to the other. There are a couple of different ways to phosphorylate ADP into ATP. In other words, there's a couple of different ways to make ATP. The first method is slower but simpler, and it's known as substrate level phosphorylation. Now think about this. Where have you heard that word substrate again? Well, substrate, of course, those are molecules that fit into enzymes. They are the molecules that enzymes work on. So in substrate level phosphorylation, what happens is that an enzyme comes along, it picks up an ADP and some other molecule with a little phosphate on it, and it transfers that phosphate onto the ADP to make ATP. The enzyme will also end up making a little product that has been dephosphorylated because it had a phosphate group removed from it. Substrate level phosphorylation, again, is slow. Each enzyme can only make one ATP at a time, but it's a pretty simple, straightforward method. Another way to make ATP that is more complicated but also more efficient goes by several names. It's known as electron transport phosphorylation. It's also known as oxidative phosphorylation, which is the name I tend to use, or sometimes you'll hear it referred to as chemiosmosis. Let's take a look at this method. This method uses a machine called ATP synthase, and this giant enzyme complex is built into the membrane of a mitochondrion. This machine has moving parts that rotate and rock back and forth, so it's much more complicated than the substrate level phosphorylation we just saw. But the advantage of using this method is that it's really fast. This little guy can take many ADPs and phosphorylate them in rapid succession. So this complex method is a way of making a lot of ATP at once.